Welcome to the webinar examining intended learning outcomes in doctoral education. This is being narrated by Bob Harris, a teacher-elected representative of the Board of Doctoral Education at Karolinska Institute and head of the Quality Assurance Working Group within that board. If you haven't already listened to the preceding webinar about the theory and formulation of ILOs, then I thoroughly recommend you do that first before listening to this webinar. Otherwise there'll be some little repetition of the basic underlying information that you're required to understand this webinar. You click each slide at your own tempo and follow the text for each slide. So let's begin. So on slide 2 you see the general aim of the webinar, which is to facilitate the learning of how to ask a PhD student questions regarding their specific intended learning outcomes within the framework of their PhD activities. And the point of this really is to elevate the pedagogical awareness of examiners or supervisors that have the task of monitoring students' progress during the PhD, either at formal moments of assessment such as half-time control or the final PhD defense, or during the continual assessment that the supervisors would be expected to do. I would like to stress that this is not a way of changing the way that we examine our students. It's just a way of complementing the type of questions that we already ask, which tend to be more focused on the science. In slide 3 you'll see a somewhat philosophical depiction of how doctoral education, or research education as I call it in this presentation, is perceived at many educational establishments. There's a clear division between what's considered to be research and what's considered to be education, somewhat like the figure in the top part of the figure of the slide, the yin-yang, representing opposite aspects of a whole. On the lower part of the panel, then you'll see on the left a completely black circle, and this will be representative of establishments which only believe in research results, publishing papers and the like. The middle picture shows a completely white circle, and of course you will not be able to conduct a PhD if you will only have formal educational moments. So neither of these scenarios is really optimal, but rather than having the very defined white and black division between research and education in the top picture, then on the lower right we maybe come somewhat nearer with the picture of the football to define research and educational activities which are linked together. As an Englishman, of course, we like the football. Slide 4 is just a reminder about the description of an intended learning outcome, what the learner could be expected to know, understand, or be able of doing, more making a judgment about within a certain period of time. If you're uncertain about this definition, then please refer to the preceding webinar shown at the bottom. Slide 5 is also a repetition just to remind you that there are 3 domains for intending learning outcomes: knowledge and understanding, that's what you should be able to know about the subject area and be able to hypothesize about. Proficiency and aptitude, what are the technical abilities that you have, the analytical methods, the presentation techniques, and lastly the ability to assess and approach, how to put your research into the context of both the scientific field but also ethically and out in the general population, to the public. Slide 6 shows a picture of how ILOs can be used in assessment of progress. And this is just to remind you that the ILOs are a way of linking both a formative assessment, where you have a continual assessment, such as would be expected between the interaction of a supervisor or group of supervisors and a student throughout the PhD education of a student, and the summative assessment, the formal checkpoints such as a half-time control and a thesis defense, where there's actually an examination form. It's important that the type of questions which we're going to be addressing are asked in both types of these assessment, and the formative assessment conducted by the supervisors will provide the basis of experience of answering such questions which the student will be expected to meet during the summative assessments by external examiners. In slide 7, we posed a never-ending discussion about whether a PhD represents a body of scientific production. Is it scientific papers and impact factors which are of importance and numbers of papers, or is it progression in learning and self-development of the individual, or indeed is it a combination of both? So let's develop that theme a little bit more. In slide 8, impact or input, you see some research results of research in my lab. I thought I'd use some examples from myself in order to illustrate this point. So you see in the first paper, which was submitted to Nature's Neuroscience, impact factor over 14. Then you see one author, a PhD student of mine, Rowan Parser, who has a middle order rank in this publication. At the bottom of the paper you see a published work, which was also representing a collaboration, where you see the same author also in the middle, somewhat higher, with a paper published in Journal of Immunology, impact factor 5.7. Now I think that most people, if you would compare these two papers, if they were included in a PhD thesis and you were examiner, would automatically assume that the higher impact factor journal then represents a better class of research than the more lowly impact factor publication. And when it comes to scientific content, and there is a reason why journals are ranked differently. However, that's the science. If you would actually think about the research education and what the role of the student has been, then you can see that in both cases the PhD student has a sort of middle author place, and you'd then need to consider what has his actual role been in both of these studies. And I can tell you that in each case the student concerned did exactly the same type of analysis. So from his point of view, the capability of learning the techniques and providing results and presenting them as part of figures in the finished publication was in principle an absolute identical amount of work. Yet most of us would still probably think that the role in the first paper is more attractive than his role in the second paper. If we continue this line of thought in slide 9, then you see the same author again, now as first author in a study which has just been accepted for publication in diabetes, impact factor 8.9, and this represents a study which was conducted on his part for 2.5 years, and a first authorship where the majority of the work he has achieved himself. This represents an incredible possibility for research education, and in all its good aspects. He's learned so many different things and been able to prove that he's learned so many different things. Yet the impact here is less than in the Nature paper. At the bottom of this slide you see another piece of work published in the International Immunology with an impact factor now down to a lowly 3.3, which was conducted by another of my former PhD students, Maya Wahlberg. In this case, even though this is the lowest impact factor of each of the four publications which are presented to you in this presentation, then this represents their ultimate doctoral education in my mind. This was a study that Maya designed herself, conducted herself, and when it was finished she came to me to discuss the results, the interpretation of the results. It was exactly the right study to do. It was in the framework of the research being conducted in my lab, but it showed me that she was a complete scientist because she knew what to do, how to do it, but still was wanting to collaborate with me for the scientific interpretation. And I think that any supervisor would be happy with this type of result in their students. However, if we would only think of the scientific aspect, then of the 4 publications here, then this might be considered, due to its low impact factor, then the less attractive of the 4 studies. So I think this quite nicely illustrates whether we put our emphasis on scientific ability or perceived scientific ability in the form of a publication, its impact factor, or whether the work involved and the learning involved actually is not reflected in the impact factor. And this is an issue which you have to consider for the rest of the presentation. So in slide 10, then you're posed with the question, how do you ask questions that address intended learning outcomes? And in order to address that question, let's click on to the next slide, which addresses a related question, how do you ask questions that address science? And I think anybody who's a scientist will have an idea about how to ask questions about science. You look at the data, and if you understand what the data represents, it's very easy to ask what the data means, how does it fit in. But that's not necessarily the first question one should ask. If one wants to address the learning of a student, then the first question that one could ask is really, how did you get to the result? And this is where you can address the intended learning outcomes of a PhD education. Slide 12 is a blank slide, and I'd like you to take a minute or two, or as long as you wish, to try and think what it is I'm wanting you to do. What sort of questions that not address exactly the science could be applicable in order to address intended learning outcomes. Try and consider what it is that you could do in that respect before you proceed to the next slide. So now we're on slide 13, and what I'm going to do in the next few slides is to show some scientific data. Now it doesn't really matter whether you understand this type of science or not. The concept, the theory of what we're going to do will be the same for any field. It just so happens that I'm a preclinical immunologist, so that's the type of, I'm going to show some data from our work, published work, and so that's the data that you'll see. But stress, it doesn't matter if you don't really understand, it's the principle. So what is it that we're looking at here? There's a long list of names on the left hand side and some fancy red and green squares in columns. Each column represents an activation profile from a particular type of immune cell, and they're different. So on the top you see the different stimulations of these cells. So each column is different, and you can see that they look different. If you look on the far right, you see a lot more red, which is showing a lot more expression of the molecules which are listed on the left hand side. And if you look at all the other columns, then you can see that the patterns vary. This is data from a so-called microarray showing expression levels of gene products. The genes are listed on the left hand side and grouped on the far left into types of genes. And red means there's a lot of it, and green means there's a reduction in the level of it. So it's obvious by looking at this that the column on the right is very different from all the others. So one can immediately start asking scientific questions about what this means, why these particular gene products induced by this type of stimulation, and so on. But let's take a step back and try and think, instead of what the data is, what does it represent? So this represents a microarray, which is a way of analyzing gene expression, but is not the only way. So there you have a question immediately. How would you compare measuring these type of gene products are there different ways of measuring gene products? And you either look at the level of RNA, or you look at protein expression, for example. So that's a question. What are the advantages of doing the assay this way compared to another? Another question that will come up from this type of study: these microarrays are commercial. You do an array. How often do you have to repeat the result in order to get valid results? This will address whether the student knows what a good scientific design is for an experiment. Do they know what appropriate controls are? How many times do they consider you have to repeat an experiment until you can be sure of the data? And so on. These are the type of questions you can ask before you start to go into what the data actually means. So let's look at another example on the next slide. So on slide 14 you see some experimental clinical data. This is showing development of type 1 diabetes in mice,